The Cannabis Conversation. A European perspective on the emerging legal cannabis industry.
Welcome to the Cannabis Conversation with Anuj Desai, where we explore the new legal cannabis industry while speaking to the professionals that are helping to shape it. Hope you're all well. Been a few weeks again. Continues to be an interesting time in cannabis. In the US, President Biden signed the Bipartisan Medical Marijuana and Cannabinoid Research Expansion Act. That just rolls off the tongue, doesn't it? Which should make cannabis research easier. So hopefully some positive steps there. In the UK, I was actually fortunate to attend an all-party parliamentary group on CBD products last week, which is led by Crispin Blunt, MP. That was at the House of Commons, which is always a fun place to go to. Slightly strange title for the group as the focus is heavily on industrial hemp and wider industries, but it was great to see so many people from the UK cannabis industry there. Guest speaker was Lisa Townsend, who is Police and Crime Commissioner for Surrey. She gave some really great insight on how varied police views are on cannabis. Some PCCs want it upgraded to Class A, which is kind of mad and flies in the face of all evidence and facts. So therefore, it's clearly a very political stance. I'm hoping to get her on the podcast to discuss more and perhaps even try and get one of the prohibitionist PCCs on too. Let's see. There was also a separate APBG on medical cannabis as well, which I sadly couldn't attend. Voltfas, who are the leading drugs think tank, presented a new report called Known Unknowns of Medical Cannabis, which is out at the moment. They talk about the multitude of challenges that doctors face in prescribing medical cannabis. And to sum it up, cannabis must be treated the same way as any other medication if we want to see mainstream prescribing in the UK, which kind of stands to reason, really. And it essentially means that the same levels of data, trials and authorizations are required for more doctors to be convinced. So more to come on this, no doubt. Once again, this episode is sponsored by our good friends at Lumino. We're talking about THC CBD ratios today, which includes a discussion on a controversial new study from King's College on the subject. Lumino have great experience recruiting for scientific and commercial roles. So anything from sales, regulatory affairs to chemists and scientists. They've recruited for those sort of roles before, and they're always happy to chat. So as always, if you need help with HR or recruitment, please do get in touch with them at luminorecruit.com. And obviously, please do mention my name when you do. Now, on with the show. Enjoy. On today's show, I have Guillermo Moreno-Sanz, who is the Global Scientific Director for Khiron Life Sciences, who are a global medical cannabis company headquartered in Canada. Guillermo, welcome. How are you today?
I'm doing great. Thank you so much for having me in your podcast, Anuj. It's a great pleasure.
Yeah, no, I'm really looking forward to this one. Where in the world are you today?
I'm in Spain. I'm originally from Spain, not the town where I live in. I live in the Basque Country in the north of Spain. It's a region that is very famous for the excellent food and the weather that is very similar to the UK. not the sunniest part of Spain I'm speaking from.
Yeah, less said about the weather, the better, I think. But thank you, man. I hope all is good. This is actually a really good topic. You and I have been sort of talking about this online for a little bit, and we'll get into that in a few minutes' time. But I usually like to start with just a bit about yourself personally. What's your background and how and why did you start looking into cannabis as a topic?
Yeah, it's funny because since there's actually a cannabis industry, I heard people saying, well, I've been in the cannabis industry or I've been in cannabis since 2016 or 2015. So, well, I've been in cannabis since 2003 when I started my PhD. I mean, all of a sudden, I have a degree in chemistry and I have a degree in biochemistry. And when I had to face a decision on what to do, professionally, doing a PhD was an option that was there because, I mean, if you want to have a career in academia, it's kind of the logical next step. But then I also figured that if I was going to dedicate 4 or 5 years of my life to research subject deeply, I'd rather be passionate about it. And at the time, I was really passionate about understanding how cannabis works. I mean, I have always thought that cannabis has a differential effect compared to other drugs of abuse and also to other medicines. That always interested me. So I decided to start a PhD in psychopharmacology at the University of Madrid, the Complutense University. Basically I started in the lab that offered me a grant to, some money to fund my research for a year. It was doing research on the subject of cannabis or cannabinoids as a drug of abuse. It was actually funded by the I would say by the DEA, the Spanish DEA, so to speak, and looking at ways of demonstrating how cannabis could be a gateway drug, right?
And was that your angle at the time?
Well, it was the angle of the lab that paid my salary, and I just tried to make the most of it. I was a very young researcher, so there's always techniques, points of view, and you are just becoming a professional of a field that you're not, an expert on. But I also had the chance to join, for example, different scientific societies. For example, this weekend we had the 22nd meeting of the Spanish Society for the Research on Cannabinoids. So at the end of the day, you meet a lot of peers, researchers, or PhD students that are on your same situation. You also, see what different approaches you can have to the topic that you're, investigating. And 3 years into this research, my PhD supervisor actually passed away, which is one of the, very challenging things that can happen during your PhD. And I was lucky enough to succeed at securing a Fulbright Scholarship. So I just packed everything, got my girlfriend at the time, and moved to California, which was a long, a bucket list dream of mine to live in California. I initially went there for a year, and eventually I stayed for 9 years. So 9 years later, in 2017, I came back with my American daughter and my American dog and my girlfriend from the time, that is my wife today, and just decided to, come back to Spain. It was seemed to be a good time to come to Europe. Spain already had, Spain does not have a medical program, but there were already several companies with licenses to produce cannabis for medical purposes and to export all their production to places like Germany or the UK. And I established myself. I decided to, transition out of academia. My last period in California was already, I was working more of a consultant to my friend. At the end of the day, California is the biggest market, in the world for recreational cannabis. I was there from 2008 to 2017. So I saw Prop 19, which was the early prop that would have made California the first recreational market, but that didn't pass for a very, very narrow margin. I believe 48% or something like that. Then it came Colorado, Washington State, and eventually California made it in 2016 on the same elections that brought Trump to the White House. So it felt like a good time to leave the country. But I was already exposed to what a recreational market looks like, and especially, what the market looks like and the marketing, because at the end, as a scientist, like everybody wants to know your opinion. That's basically what I started doing, just telling people, my opinion about things. And then you realize that nobody really has a background from which they can distinguish, what is true from what is not. And there's a lot of people getting away with just selling smoke in the industry just because nobody knows any better. From that perspective, I always had, an advantage because, I know the science of it. And, it turned out to be useful for some people. When we said about how companies don't invest in research and stuff, there's so much more that is technical, that is science-based in a space like this, beyond the efficacy and safety of clinical trials. So I started working as an independent consultant. I always had friends in the industry. And eventually, right into the lockdown, Khiron, the European team of Khiron was putting together a team to set up operations in Europe. And they were looking for a scientific director. So I started as medical and scientific director in Europe, especially taking care of our relationship with drug science and T21. And eventually, since I was the only team member in Europe that was fluent in Spanish, I eventually made my way to Colombia, where the main headquarters of the company are. I met, all the colleagues at the time. And, we're present in Peru as well, Colombia, always, Mexico and Brazil are markets that, we're entering slowly. So, I eventually transitioned to this global position in which I oversee actually the scientific production of the company. So, to me, it really is a dream come true. When I think about why I decided to start, in this career, the fact that Spain is looking at now, setting up this program, and I have the opportunity to, talk to the professionals that, are doing so and participate on the process, I feel really accomplished. So, I'm extremely happy, to have this position. You never know what's going to happen next, in this industry, but so far it has been, a very nice ride.
Yeah, thank you. That's a brilliant story, and there's loads to that. When you started out, 2003, obviously you're a heavyweight veteran then in that case. And particularly if you were studying kind of cannabis as a drug of abuse, could you have ever sort of envisaged this roadmap that happened? Like not just being able to do it in the US, but now being able to do it in Europe and globally?
Well, the thing is that, at the time, there was already evidence. There's a long record of evidence for the medicinal properties of cannabis and cannabinoids. But the science behind it was fairly new. The endocannabinoid system was only discovered in the 90s. And it was a very, I would say, fast-paced discovery. From the time we discovered the first piece of the puzzle until we figure out pretty much the entire dimension of it, it went only, it was a decade. It was basically the 2000s. So when I was a bit frustrated about, having to investigate cannabis as a gateway drug when, this was clearly propaganda that was used in the 30s and the 40s to ban the use of the international trade of cannabis. But as I'm saying, at the end of the day, I was still, learning techniques on how to investigate neurotransmission, how to investigate behavior in animal models. So those were still skills that, were going to be very useful for me in my career. Obviously, I much rather, prefer what I did in California later on. I was actually developing new drugs that target the endocannabinoid system. These were, the first, I would say the 2.0 version of cannabinoid drugs. The initials were copycats of THC, so molecules that activate directly the CB1 receptor. And the 2.0 that I was working on were drugs that inhibit the degradation of anandamide. So in a way, if you think about it, from a pharmaceutical standpoint, it's equivalent to the inhibitors of serotonin reuptake, for example, which are the antidepressants that we have today in the clinic. So it's a different strategy. It's more, it's assumed to be more selective, with less side effects. So more of the likes of the rational drug development of the early 2000s, so to speak. Now we have a different approach. We understand that molecules that have one single target, are more limited on their application. And as a whole, I think the pharmaceutical field is, more open to explore more pleiotropic molecules that are more dirty or promiscuous, such as, CBD, for example, that don't have a one single target, but that can, have many different activities in the organism. So in that way, I got also impatient, so to speak. We were having excellent results in animals. We had a molecule that was very promising that, could hit clinical trials very quickly. 6 or 7 years later, we were not even close to get to the market. And I decided that phytocannabinoids were what people and patients were already taking. And it was very worth investigating what was the outcome of these patients using phytocannabinoids rather than waiting 20 more years before we could have, a drug in the market for a single indication that will jump over all the loops that traditional clinical development will have. So in that sense, I think that, I've been able also to move from, between fields until, at the end of the day, there was an industry for me to land on different than the pharmaceutical industry.
Yeah, that's a great journey. And it takes us quite nicely to where you are now, actually. Perhaps you can tell us a bit more about the stuff you're doing at Khiron.
Yeah, Khiron, we define ourselves as a global cannabis company. What does it mean is, Khiron, we just made 5 years. So initially the company was based or is based in Colombia. I don't know if it was the first. I mean, I read it was the first, but was one of the first movers to obtain license for the production and manufacturing of cannabis medication in Colombia. And we are also a public company in Canada. So we are in the stock market in Toronto, which also adds an extra layer of control and surveillance by, different agents, which makes me feel very safe about what I do. Because, I mean, at the end of the day, not everybody is willing to have both feet on the white side of the of operations in this industry. And that's something that you learn very early too. So, I mean, the proposal of Khiron was very attractive from that standpoint. And at the end of the day, I think we have been very realistic with our final goal, which is to improve the quality of life of people. And to that end, we need to increase the access on medicinal cannabis. The key to the access are doctors. So what we had to do, or what my responsibility was to design the communication with doctors. How do we train doctors? How do we explain what cannabis is? How the patients can benefit from cannabis? Every jurisdiction is different. The products are different, but the main goal is always the same. So that's something that has helped us also expand into Europe, because, the message is very alike. The products might be different because the flower is allowed in Europe, it is not in Latin America. That makes profound differences in, for example, the average patient that we see. I mean, our patient in Colombia is a 60-year-old female. In the UK, it's a 40-year-old male. In the UK, they consume high THC flower. In Colombia, either, balanced or high CBD oils. So it is really different, but you see how doctors in the real world are making a difference for these patients, right? I mean, obviously, it won't work for everybody. That's a given, but still we see good adherence to the treatment. In general, legislations only allow patients that have already failed some treatment, so they're refractory to treatment, to take cannabis. So, there are a number of challenges, but as first movers, our job is to face them and to try to solve them. So in a way, sometimes you think you're just paving the way for more people to come. At the same time, sometimes you feel that nobody's going to bother to come this way, because sometimes it's really hard, right? And there's a lot of things that you don't know, and even the people that need to give you answers have never faced these kind of challenges. We were, for example, the first company to export live cuttings from Colombia to Europe. So it was the first phytosanitary certificate that the Colombian Institute of Agriculture ever did for cannabis. So, it is always a challenge, and having, good colleagues, a good team, it always helps. But at the same time, there are just things that just because you are the first one doing it, but at the same time, I can't complain. This is what I really like to do. I like challenges. I like doing different things. I like every day we're facing different stories. Particularly in the UK, Khiron was one of the founding members of Project T21, which is, for the listeners that don't know T21, it's a multi-center registry of patients in the UK that receive treatment with CBMPs, with cannabis-based medicinal products. We provide subsidized cannabis medication for those patients that enroll in T21, and they grant access, or they grant drug science the ability to conduct research with their data. So we just published a paper with 344 patients in the UK medicating with receiving our high THC flower or chemotype 1 flower to treat several indications, mostly chronic pain and anxiety-related disorders. But, there's many, there's up to 3,000 and counting. This is still very limited. We were hoping, T21 comes from 2021, as in 20,000 patients by the end of 2021, right? Obviously, we didn't meet that goal. It is work in progress. It's really challenging to get doctors to, out of their comfort zone, out of the way. Fortunately, we have found some passionate professionals that really, take pride on making, their patients feel better and have a better quality of life. So we're always, aiming at supporting those doctors.
Yeah, fantastic. Great project 2021. I've had David on here a couple of times to talk about it. I mean, I totally agree. It's not the bottleneck, but one of the keys to unlocking it is doctor education, really. And I guess one of the things that will help convince them is more compelling evidence. And it's very interesting what you said. Maybe commercially being a first mover can be a good and a bad thing sometimes. But I would imagine as a scientist, and certainly I get this whenever I speak to scientists, that looking at new things for the first time is a particularly attractive thing for people studying. So I totally get that. So I really appreciate your story there and all of the things that you're up to. I think it takes us quite nicely to the meaty topic we're going to talk about today, though, which is how important are THC CBD ratios. There's lots of aspects to this. And, I think there was some semi-received wisdom around this. And then there was a King's College study that was released just recently, which has maybe caused people to have a slight think in a different direction. We'll come on to all of that in a second. Maybe we can talk a bit about the basics before we get into the interaction between the two cannabinoids. So maybe we'll start with THC, the most famous. How does THC, on a very basic level, don't need to be a full-on biology lesson, but how does THC work on the body?
Yeah. So THC indeed, is the main cannabinoid. I mean, some people might say it's the only cannabinoid. I mean, I don't want to be too controversial, but at the same time, I really think that CBD sometimes steals the thunder of THC, meaning that we didn't know about THC until, around the 1940s, probably, was the first time where we were able to study at a molecular level cannabis. It was LaGuardia report. Fiorello LaGuardia was the mayor of New York at the time, and he was a firm opposer of Aslinger when the prohibition was coming into place. This was 1944. And, if you read this report, there is particularly the lead psychiatrist or the lead psychologist, Flores Halpern, that describes what the effect of THC is in humans. And since then, all the substances that were able to elicit these effects, both in human and animal, lab animals, mainly rodents, mice and rats, were called cannabimimetics, meaning that the effect of THC was for the longest time associated with the effect of cannabis. Also, because if you remove THC, then the effect of cannabis, the cannabimimetic effect, is gone. And it actually stems back to the early pharmacopoeias, and I'm talking about, the late 19th century and early 20th century, because this was way before people know about THC and way before HPLCs or chromatography or any means to measure the active ingredients were prepared. In the pharmacopoeia, there were instructions for pharmacists to prepare cannabis extracts. And to determine the potency, they had to use 2 dogs. This is what the pharmacopoeia reads, right? So take a grain of rice of the cannabis extract and give it to a dog. And depending how motor impaired the dog becomes, then you know if it is too much or too little, and you have to adjust the concentration. And this is the effect of THC. So from the very beginning, THC is the main cannabinoid, is what defines the effect of cannabis. So later on, we learned that THC, because it is a lipid, people was not sure in the 1990s. So it was a surprise, the fact that THC binds to a particular receptor. Because being a lipid, usually, it was assumed that it will get, mixed into the membranes of the cell. They will, may change the fluidity of these processes, things like that, but not that they will have a selective receptor. So discovering a selective receptor for a lipid was critical not only for cannabis, but for our understanding also of lipids as signaling molecules. Because when I studied biochemistry, lipids were building blocks for cell membranes and stuff that is storage energy. So basically fat. But it was not, we didn't know yet that lipids could actually be messengers and communicate cells with each other. So in a way, the endocannabinoid system also changed a lot of that. And the fact that we discovered the endocannabinoid system after THC, it really defines the endocannabinoid system as, the target for this particular molecule. So basically, THC is a partial agonist of cannabinoid receptors. This means that activates these receptors, but not so much as synthetic cannabinoids that are infamous synthetic cannabinoids such as K2, spice, all these cannabinoids that we hear about, zombie outbreaks in Manhattan and all these issues with this full agonists, meaning that these are way more potent activators of this receptor. So actually THC mimics better the way our body engages with cannabinoid receptors than other synthetic agonists, right? So THC, it's the only molecule that we have in the clinic to activate these receptors. There's no other molecule. Maybe acetaminophen or paracetamol can be modified into a molecule that does this, but it's the only molecule. So CBD does not do this. And in that way, THC is very unique.
I mean, there's loads of stuff there around the synthetic issue, but sticking to THC, when it activates the receptors, what are the sort of typical effects that we experience? I mean, I know the stoned effects, etc., but from a biological perspective.
So from a molecular perspective, you have a couple of immediate effects. In general, it closes calcium channels. What does it mean? It means that neurons cannot fire so often. So in general, activating the endocannabinoid system has an inhibitory effect on neurotransmission. However, this system is not present only in neurons, so it is present in many other types of cells. So we know that activating CB1 causes analgesia, reduces body temperature, affects locomotion, equilibrium, balance, but also we know it's gastroprotectant. It really protects all mucosas in the body. And, it helps regulate the energy balance. Also, this is one of the reasons it causes hypothermia. And in general, it engages on physiological responses that deal with bringing the organism back to basal state or rest state. And this is what normally people say about maintaining homeostasis, meaning that the body is never still. I mean, there's always reactions going on. So the equilibrium is always active. It's just about having as much stuff in one side of the scale as in the other, but it's always changing, right? So the endocannabinoid system, or THC in particular, through the activation of CB1, the result is bringing the body back to rest state or normal state. So it increases appetite. Well, I mean, the traditional effects that you see when people consume cannabis, right? So these are all responsible. I mean, THC is responsible for all these effects.
Right. This is so interesting. And wasn't what I was going to ask you originally, but while we're here talking about it, that synthetic versus phytocannabinoid issue is something that's talked about a lot. We've talked a bit about the Entourage Effect on the show and, the evidence or lack of, or, that it's still something that's being studied actively. Could you sum up very quickly your ideas on, is it just a pure overload of a receptor with a specific isolated compound? Or
we need to clarify one thing. When I talk about synthetics, the difference, what you're referring to is the difference between a phytocannabinoid and a synthetic version of the same phytocannabinoid. When people say that, for example, synthetic THC or synthetic CBD do not have the same effect as, THC and CBD coming from the plant, because in general, we work with full spectrum extracts. So there are, other molecules coming from the plant that nuance and fine tune the effects of cannabinoids. This is one thing, not the one that I was referring to. When I was referring to synthetic cannabinoids, I mean other molecules that activate CB1 receptors, the exact same receptors, but not in the same way THC does, because THC is a partial agonist. So if you think about it, let's say that THC can turn on the volume dial up to 5 out of 10, right? So it doesn't matter how much THC you take, the volume will only go to 5, sorry. But if you take a synthetic cannabinoid that is a full agonist and they can turn the volume up to 10, then the effect will be different than the one that you have with THC, even though you are activating the same receptor, right? The other discussion comes with, okay, the molecule is the same, but you have a phytocomplex that might be doing other things. So in that case is when we talk about the entourage, and that's a completely different conversation that we can get into, but it's not what I was talking about.
Yeah, no. That's a different conversation for another day. I did quite like the canine calibration that you talked about, having dogs as the kind of testing regime for how strong something was back in the day. Very interesting. So, like, while we're on THC, high THC is very popular both in the adult use and the medicinal markets, and in some ways, the medicinal market follows the adult use market in some ways.
Yeah.
What are the sort of benefits and risks that you see of the high THC products?
Well, I mean, I understand is what we normally talk about, but I really like to talk about chemotypes because high THC, I mean, the amount of THC in the product is not that relevant, meaning that, people immediately assume that high THC means that you are getting a lot of THC. It means that the concentration of THC is much higher than the one of CBD. And this is where we will eventually get to the ratios, right? So people call it unabated THC or, high THC. But the thing is that the dose that you're getting depends on how much you're taking of it. What it means is that you're getting way more THC than CBD. And in terms of products, in Europe, we have both flowers and extracts, right? So, for example, Germany has been using dronabinol, which is the word that we use for pharmaceutical THC, even if in the US, it is synthetic. In Germany, it comes from extraction and purification of cannabis, of the plant material. But it has been used with quite success for a very long time. And, in the flower format, the same thing. We know that the concentration or the amount of THC or the content of THC has been increasing over the years because it has been selectively bred in for that reason. So, we can fairly speculate that the lower the level of THC, the higher the ratio with CBD will be, because, at the end of the day, if you keep it one constant, then you're breeding the other one. Eventually, you're switching this balance. So, in general, from a pharmacological standpoint, THC is the molecule that you're interested in. And, there's a lot of curiosity, to use that word, about, how much safer, it becomes when you add CBD to the mix. And this stems from literally, and you can ask Ethan next time you have it around, this stems from the development of Sativex. Sativex was the first drug that was approved by a drug agency. And it's a product that has equimolar or equivalent concentrations or amounts of THC and CBD. There's several reasons why one will want to do this in 2001. One of them could be that these being natural products and natural molecules, they are very hard to protect and to patent. So when you work with natural molecules, one way of overcoming this limitation is to work on a composition or to work on a formulation. And Sativex is an example of both. You are combining two cannabinoids that do not necessarily come together in nature, because actually Sativex was made with two different extracts, one extract of high THC and one extract of high CBD, that then was mixed. And with a particular formulation, which was an ethanolic spray, right? So it was very difficult to reproduce. And that it gave GW, enough, a robust case to patent that product and therefore to conduct clinical trials and to further develop that product. And since then, this notion started of THC alone being less effective and having more side effects than the combination of THC and CBD. From there stems the fact of the entourage. For the longest time, especially since Ethan in 2011 came up with the British Journal of Pharmacology, the taming THC, the case for the particularly the terpenes to modulate the effect of THC. Many people were talking about the entourage effect and the combination of molecules, but at the end of the day, the examples that they will give you will be the relationship between THC and CBD. To the point that today, when we use chemotype 1 products, a lot of doctors that have read all this literature come and say, well, but we're being told that you need CBD in order to reduce the side effects and especially the unwanted side effects of THC. And the truth is that, or the experience that we have is that in general, one-to-one formulations are safer, meaning that patients report less side effects, especially orally administered, and that for some people, THC works alone works just fine, depending on the indication that you have, especially if you have nausea and vomiting related to chemotherapy, or you want to increase your appetite, or you have neuropathic pain. I mean, for these things, we know THC works better than CBD. And in general, THC and CBD in combination work as well as THC. However, just to be safe, since we have this notion that, a combination of both, is going to be less aversive than THC alone, that's why, people tend to use balanced combinations. Because, we really don't know any, the design of the clinical trials with Sativex were designed to prove efficacy and safety of the combination. Some of them will actually compare the combination with THC alone, seeing, for example, that Sativex was slightly more effective than THC. So, maybe CBD also has a component to it. But in any case, the amount of CBD that you're taking is usually very low compared to the doses of CBD that you will take when you take CBD alone for those indications that it's recommended for, which is in general pediatric epilepsy.
Yeah, wow. I'm not sure where to start with that. There's so much there. Well, we're going to talk about the King study in a second.
Yeah.
Let's just quickly talk about CBD working on its own.
Okay.
Not to say the efficacy of any products that are available over the counter, because, there's a wide range there. But in theory, how should CBD work? And is it anti-inflammatory, as I understand it?
Yeah. So, CBD is what we call, it's a dirty or promiscuous molecule, meaning that it has a lot of different targets. Some of them are within the endocannabinoid system or the expanded endocannabinoid system, and some of them have nothing to do with cannabinoids. So, we know CBD is a negative allosteric modulator of CB1. That means that it binds to the same receptor as THC, but in a different pocket. So this was actually perceived as the explanation why CBD will, nuance or fine tune the effects of THC, especially those that are aversive. Today we have studies that have shown also that CBD might also reduce the efficacy of THC through this very same mechanism. But again, this is all research that, it just brings more pieces to the puzzle, but none of it, it's a killer experiment or the defined answer to what we're looking at. So we can say that CBD is really, it does not activate cannabinoid receptors. That's why I was saying at the very beginning that it really is not a cannabinoid. It's an active molecule that is in cannabis, but it does not activate the endocannabinoid system the same way as THC does. For the longest time, it has been described as a FA inhibitor, which is the enzyme that I studied for many years, meaning that it can inhibit the breakdown of anandamide and therefore raise anandamide levels and indirectly activate CB1. In my hands, I never seen that. There's a study from 2008 in which psychotic and schizophrenic patients will take high doses of CBD, 800 milligrams a day, for several days, and then they had increased anandamide levels. And that's one of the reasons why, we know chronic administration of CBD might do something to endocannabinoid levels, but we are not sure exactly how. So as a pharmacologist, that's also, one thing that I don't like about CBD, that we don't have a clear mechanism of action. It also activates 5-HT1A serotonin receptors, but also in concentrations that did not seem, attainable with the doses that people usually use. And don't get me wrong. Most of our patients in Colombia are taking chemotype 3 or high CBD products. But there's two things that I think are essential for these kind of products to work. One is accumulation. Typically, CBD does not work overnight. You need good 3, 4, 5 weeks, after a month, you see the effect coming up. And this is also true for Epidiolex. We've seen that you need a titration period in which you increase the dose little by little. And these are fatty molecules that are going to accumulate on your fat, on your brain, on your cell membrane. So accumulation might have to do with the how CBD functions. And also, these are full spectrum extracts, meaning that there is some THC on it. So by the time you're taking 30 to 50 milligrams of CBD, you're probably taking 2 milligrams of THC a day. 2 milligrams of THC a day might be an active dose if taken every day, especially on the elderly. So that also speaks about, I mean, can you achieve 2 milligrams of THC with a high THC product? Of course you can. You know, you won't have the effect of the CBD, whatever it is, this rounding or protecting effect of CBD. But that's also what, you know, the doctors tell you. You know, they see how people, especially, you know, at an older age, they're way more sensitive to THC than younger people, for example. And they prefer to have products that have more CBD so they can little by little, you know, reach doses of THC that are effective, but, you know, with a much more safety profile.
Yeah. God, this is fascinating, isn't it? Obviously, there's a huge CBD market in the wellness market.
It is.
But, you know, by and large, those will be broad spectrum, i.e. without any THC. So,
yeah, I mean, in general, but that's the thing about cosmetics or the wellness is that you really cannot make any health claims, right? So as long as there is no health claim, then everybody has their own reasons for doing that, you know, and I mean, this is also, you know, an entire topic for another podcast, but, you know, there's a couple of publications out there saying how expectations and the expectative of the consumer, it's critical for the reported efficacy of CBD, which is another way of talking about the placebo effect, right?
yeah.
But we know that cannabinoids are, you know, very involved into the placebo effect. So people that have, for example, some mutations in their endocannabinoid system might be more easily, you know, to suggest or to self-suggest to the effects of therapy. So, I mean, I'm not saying there's nothing to it, but, you know, cosmetic and wellness are, you know, fields that you really don't need to prove anything.
Yeah, the Wild West, let's say. But and so again, the superb summary of lots of different aspects there. And, you know, I think, like I said, I think the conventional wisdom was sort of maybe a balancing act between THC and CBD. You've explained that it's a bit more complicated than that, obviously.
Yeah.
And then this new King study came out a couple of weeks ago. You know, first, maybe you can very briefly summarize what that said, and maybe you can give a bit of analysis on what you think the value is in that study.
Yeah, I mean, I really like the study because, I mean, I think that it really addresses the research question. Being the research question is inhaling different ratios of THC to CBD safer than others. So, and, you know, they did a very neat design in which they have THC alone, THC in a ratio 1:1, and CBD in a ratio 1:2 and 1:3. And they actually got all this cannabis from Bedrocan. So they actually mix and match to make the ratios. And they even got placebo cannabis that Bedrocan has. And they added it to make sure that all the pods for the volcano had the same weight. So, I mean, it's commendable the effort they did to homogenize the conditions. But it's also fair to say that this is a human lab experiment. What does that mean? It means that these are not patients. These are volunteers that are investigated in very controlled conditions. So, I mean, for an experimenter, it's a dream come true because, I mean, I have done experiments like this in mice and rats, obviously, you know, and there's so many things that you can do to animals that obviously you cannot do to humans. But human lab studies have really brought a lot of knowledge to what we know today about the endocannabinoid system. But the limitation is that you cannot always extrapolate what you learn in human lab studies out of those control conditions, you know, and indeed the results are, you know, contradict common knowledge. And that's always something that is very interesting. But at the same time, it's a challenge to explain why this is happening. Why, when your results contradict the literature, it's always a pain in the ass. Because, you know, but that's the good thing about having such tight control over your experiments. You know, I mean, you can be very assertive within the conditions that you have created, right? So it's funny because there were another couple of papers from last year that I wrote a piece about, and I was familiar with literature precisely for that were saying indeed that chemotype 1 or THC predominant flowers could cause way more anxiety and stress and even, you know, paranoid thinking. I'm not saying psychotic attacks or crisis, but even in experimented consumers. And, you need to go deep into the design of these studies to understand what's going on, because, some of them are done. I will put an example in New Mexico, right? Data is collected with an app in the phone. So the King's College study, they use validated scales, and they use some tests, like they give you, a couple of pounds, and they send you to a vending machine that is on the other side of the hospital. And then this is something that typically creates paranoia in people that have consumed cannabis. But at the same time, even from the selection criteria, like who do you investigate? Who is allowed to be in the study and not? So their population is extremely young. It's incredible. It's like 26-year-olds is the average. And this is all people that had consumed cannabis in the past, but has not continued consuming regularly. So this already tells you about what is the relationship of cannabis with these people, right? And maybe you are just selecting certain individuals that are going to later on skew your results. Again, you need to put the cat somewhere, right? So for this, I will say that for non-regular consumers, for people that are either cannabis naive or have tried cannabis, very few times, these results might apply. And the results say that it doesn't seem like, the amount of CBD combined with THC will have any effect on the, I don't want to say psychotic-like effects, because at the end of the day, these are healthy volunteers, subject to a battery of tests. So obviously you are measuring the propension or the divergent thinking that it's created, or whatever, or the psychotic-like feelings that these people are having, but obviously you are not inducing psychosis in any of them. These are not patients that have diagnosed condition or comorbidities, or that are taking other medications. So it's very hard to extrapolate to a medical cannabis, scenario in which you have, mostly people that have already a history of self-medicating with cannabis. And maybe in this population, in patients that are used to THC predominant cannabis, other researchers have found that actually, having a 1-to-1 ratio or having some CBD on those products is going to make a difference towards, being less anxious or less all these side effects that they're describing. And the truth is that they see THC eliciting some of these effects, while there are others that in their hands, THC is not creating any disturbances in those tests. So, I think that you always need to take, in general, my advice as a scientist is that you always need to take research with a grain of salt. Titles and headlines from non-specialized press, can always carry a message way farther than it was initially intended. It has happened to me too. Unfortunately, not everybody can read and interpret, a scientific paper. So you just need, is what we're discussing. This is, I think it's a nice piece of work. I think it brings information that is really valid, especially for people that is naive to cannabis. And the readouts of the authors, mainly in terms of public health, it's more about, it's a warning about, don't think that, cannabis that has more balanced ratios between CBD and THC is safer. In general, what is safer is to consume less THC.
Yeah.
As an absolute amount, not in relation to CBD.
Yeah. And I think that's the nuance, isn't it? And again, there's so many good topics that come off the back of this, but, I guess one of the things is the accumulation point that you were making earlier. If you have a infrequent / cannabis naive patient base that you're performing these experiments on, maybe there's a slightly more dramatic effect. And I guess what it ultimately says is that we still are very early on all of this research, like so much more needs to be done to kind of make the mosaic.
Yeah. And because, doctors tend to read this very literally. And when I tell doctors is there is more variation in your patient than in the cannabis he's taking or she is taking. All patients are different. We need to standardize them to investigate them. But the reality is that doctors see patients one by one. So there are, not all endocannabinoid systems are equal. There will be people that is more vulnerable, that is more sensitive to THC. And in those cases, some of them might just need to stay away from it. Some might actually tolerate it better when you add CBD to the mix.
Yeah.
But, it really goes on a one-to-one case.
Well, this is the sort of, the vanguard of personalized medicine, which is a super interesting aspect of cannabis research. So look, as we get towards the end, and we could talk for loads longer, it feels like THC CBD ratio is quite a reductive, metric, if you can call it that, in order to just a very simple kind of guidance tool, let's say. Where do you see its value? And if it's not too huge a question, how important are terpenes and minor cannabinoids to add to this? Balancing out the need to be able to convey information in a simple way.
Yeah. I think in general, we are reaching an agreement or a common understanding about chemotypes. I think this is helpful information. Chemotype 1, predominant THC, chemotype 2, balanced THC CBD, chemotype 3, predominant CBD, because it might help also physicians to understand what indications can benefit from what. And why do we, for some things we use just THC or a balanced combination. In general, I don't think we have enough knowledge about minor cannabinoids. This is a clash of things. First, we have, what products do we have available? It's very rare that you have minor cannabinoids as a lead cannabinoid in a product. From a pharmacological standpoint, we know very little about them. We know, because also, we apply the same test we apply for everything. So there hasn't been, systematic research for every single minor cannabinoid. But honestly, it's been several years that we modified the structure of THC to make new molecules, new synthetic molecules. And we know more or less what, THCV, CBDV, THCA, CBDA, assuming that these are not minor, right? But CBC, CBN, and none of them have an impressive pharmacology. I will go back to THC is the only molecule that we have in the clinic that activates cannabinoid receptors. And to me, there is so much that we don't know about that already that, I don't see why we should be investigating others. I have some colleagues that tell me, but you haven't tried them, right? Because if you try them, it's obvious that they have, a very impactful effect. So yeah, no, I haven't tried them myself. Maybe I should start doing that. But I think that we have enough unknowns with what, we have right now. In a way, the same goes for terpenes. As I told you, I was in California in 2015 through 2017. So I saw the whole rise and fall of the, I would say, mood specific cannabis product development. This whole notion that this is your cannabis for focus, this is your cannabis for relax, this is your cannabis for appetite, this is your cannabis for exercise, this is your cannabis for after sex. So all this nuance experience of cannabis based on a particular combination of terpenes, I think is something that helped create very colorful labels. But honestly,
branding and marketing.
what I say on Khiron's, medical education is that it's true that cannabis varieties are different, and that the psychological and the preference of patients and consumers might differ. Obviously it's a natural product and they are different. It's not, you can have 10 different apples and 10 different people, and every person will prefer an apple, right? So with chemotypes, it's kind of the same. If you need a chemotype 1, you might prefer one variety over the other, but start assuming that one will be more efficient or more efficacious or more safe because of the terpene profile might be a little bit of a stretch. But still, preferences help adherence to treatment. So, if you have a bad experience with a cannabis flower, doesn't mean that a different cannabis flower with the exact same content of THC might not have a different effect on you. So that's why we recommend people trying, 2, 3, 4 varieties until they find, what works for them. In particular, I talk with my lead psychiatrist often because, he likes to prescribe sativas for the day and indica for the night. And I'm always like, man, this is so 2010. It's like, why do you do this? And he's like, no, but some varieties are more narcotic. They're more relaxing, that they do the cat's look. And it's true that even if it is true that some varieties do have that effect, it is impossible to decide ad hoc to, just because it's an indica or a sativa, that doesn't give you that information. So, I mean, I definitely see a value on the chemotype. I mean, I think that's a useful tool for prescribers. And then, embracing the variety of cannabis and deciding that, well, I mean, it's the same with paracetamol. Like my wife cannot take it in pills because, he prefers soft gels, or I prefer, a powder that dissolves in water. So, I mean, this is just, it comes to individual preference on an API, on an active ingredient. So assuming that you have your THC, your active ingredient, or your chemotype 2 flower or whatever, then it's licit for patients to have a preference on flavor or aroma or nuance effects. So that's one of the reasons why, I mean, the broader the portfolio that patients have access to, and, their reproducibility, because obviously, whenever you have many, different products, I mean, that comes with a toll that, not everything is always available. So, I mean, people are forced to change from one to other, and then is when they realize that not all varieties are created equal. So, it definitely, that's something that affects, but I don't think you can decide ad hoc by the composition of terpenes what the effect of this variety is going to be on a particular patient or not.
I love this. It's a really good way to end the show, actually. And I kind of, what I take from that is, the chemotypes is a good kind of general indicator for physicians. I guess the terpenes and the X factor of the minor cannabinoids, I like to describe it, because you can't really, we don't know enough about any of them to really, ascribe specific benefits. But I guess ultimately, if it's more likely to keep you taking that thing and you feel more relaxed taking it, then maybe you'll get the therapeutic benefits from it more easily than you can with things that you dislike. So yeah, and again, with all of this, more research is needed, and we look forward to that. Guillermo, we could have talked for a lot longer, man. This is super interesting, and there's loads of sort of spin-off topics that we could do. So I'd love to have you back on at some stage.
Yeah, anytime. My pleasure.
Cool. Nice to talk to you, man. And then I look forward to speaking to you again soon.
Excellent. Thanks for having me, Anush. That was great.